I'm Virginia Tippy, and I am the co-chair of the EYC Environmental Committee. And I want to say, first of all, really welcome so much to this very special EYC Earth Day presentation. You know, it's really hard to believe that it is the 50th anniversary of Earth Day. A lot has happened since 1970. Think about it. And literally, we've seen a major increase in environmental awareness, but there's still so much more that we need to do. Now, as I mentioned, I am the co-chair of the EYC Environmental Committee, but I'm also an oceanographer and a former federal manager. And in my world, my professional world, our speaker tonight is truly a superstar. She is not only a superstar for her scientific work, but she is a superstar for her leadership at the national and international levels. Dr. Rita Colwell is a distinguished professor at the University of Maryland and Johns Hopkins University. And there's a reason she's distinguished. She's a renowned environmental microbiologist and also an oceanographer. And she was the director of the National Science Foundation, and under both President Bill Clinton and President George W. Bush. At the international level, she's held many advisory positions across the globe in every country you can imagine. And most significantly, she developed an international network to address infectious diseases and water issues in both the developed countries and in the developing countries. And this important network is known as the Safe Water Network. And I can tell you, she's in the center of all the action of infectious diseases. And we're really pleased to have her. But you know, you have to recognize that Dr. Colwell, she's been around. She's got numerous national and international awards for her work, including the National Medal of Science, which is presented by the President of the United States, and the Stockholm Water Prize. Think of Nobel Peace Prize. It's the Stockholm Water Prize presented by the King of Sweden. But she's also a knight of the Legion of Honor in France. So I guess we should call you Knight Colwell. She even has a major geological site in Antarctica named after her for her work in Antarctica, known as the Colwell Massif. It's a mountain, essentially. I also think it's very relevant today that Dr. Colwell led and co-authored the American Academy of Arts and Sciences 2019 report, Science During Crises, which was a call for action to the governmental agencies and the non-governmental organizations to increase the role of science in responding to disasters. What a timely report given the COVID-19 scenario we are seeing now. And finally, for you sailors out there, Rita's an avid sailor, and she's been a member of the Severn Sailing Association since 1970, isn't it, Rita? You've been with the Severn Sailing Association for a long time. She's not only studied the bay, but she's also sailed it for many years. And I assure you that Dr. Rita Colwell cares deeply about our bay, our oceans, and this very special planet that we call Earth. And tonight, Dr. Colwell is going to talk about the link between climate change, the oceans, and human health. And she's going to bring it home to the Chesapeake Bay and to all of us. At the end of her presentation, I'm going to be asking Dr. Colwell questions from the audience. So please direct your questions to me via the chat box. So you just need to go into your chat box on your screen and send me a question, and I will then direct it to Dr. Colwell. And it is truly my honor to present Dr. Rita Colwell. Rita.
Thank you so much, Virginia. It's a great pleasure to have the opportunity to speak to not just the Yacht Club, but I guess Greater Annapolis. That's wonderful. John, is my voice coming through okay?
Sounds very great.
Good, okay. So what I'm going to do this evening is take you on a kind of a long tour through the Chesapeake Bay, but also to Bangladesh and some other parts of the world. Then what I want to do is to go through a focus on water and to demonstrate using a disease, cholera, which I've been studying for the last 30 years, and to be able to explain to you the distribution of the disease globally and use that as a paradigm as I move into some genomics work and then explain some recent studies on COVID-19. So I'll talk to you about cholera, which is a global disease. And then, it's an acute water-related disease. And, it's not pandemics. And the pandemics been recorded let's say about 700 years ago. We don't know what the pandemic was started in 1960. The disease in about 50 countries affects us in the unfortunately, not to cholera and it's kind of here because years ago, and also Washington, D.C. Washington, D.C. was considered a miasmic swamp. Well, some people think it still is, but for different reasons. Now, the bacteria exists naturally in the aquatic habitat. What I'll describe in a moment is the discoveries we made in the Chesapeake Bay, showing that the bacterium that causes cholera is in the Chesapeake Bay. So you eradicate it because so if we don't have these bacteria in the environment, essentially the Chesapeake Bay would be, shall we say, paved with crab shells. Now, this gives you a slight picture, just a very brief picture of how we work with the organism in the laboratory. You see the petri dish with cholera bacteria having been streaked along the surface of the agar, and these yellow colonies are actually numbers of bacterial cells.
Dr. Colwell,
Yes?
I just wanted to say there seems to be some background noise, and I think you might be accidentally hitting your laptop or your computer while you're speaking. So just wanted to give you a heads up. Sometimes you're clear and then sometimes it breaks up.
I'll have to tell the audience that this is the first time I've given a lecture sitting in my kitchen using a laptop. So bear with me. Give me a break. So the bacterium is rod-shaped. It's a little curved rod, as you can see. In fact, that's why it's called Vibrio. It means curvature in Latin. And it has a little tail that propels it through the water. The cholera victim is exactly as you would see it today in the cholera hospital in Bangladesh. We collect the fluid lost, and that is, we attempt to replace it with oral rehydration. Now we see it in Chesapeake Bay. You see the numbers up and down the bay. Those are stations that we've been sampling for the last 30 years on a regular basis and gathering data. The work is funded by NIH and NSF and also the state of Maryland. Very, very delighted that we've been able to work so well with the Department of Health in Maryland. Now, this is essentially what was one of the first discoveries. This was made about 30 years ago. We discovered that vibrios are associated with plankton in the bay, and that in the summer months, they're very abundant. This is important because one of the kinds of species is omnificus. And that's a pretty bad bacterium. If you have an open cut and you get exposed to it when it's in the water, usually down in the lower bag, and enters your system, it can be fatal. In fact, the fatality rate is about 50%. It's a very tough bacterium. This is the copepod in the bay under an electron microscope. You see the egg case to the left. The tend to attach to the egg case and probably have evolved a relationship because they have a very powerful enzyme. And the copepods just kind of discharges the eggs into the water column. And we think the Vibrio probably, in its association with the copepod, plays a role in the life cycle of the copepod. So its host is one that it has developed a long-term historical relationship. Now, over the last 30 years, 40 years working in the Bay, we developed an understanding of the relationship of the bacteria to the natural flora and fauna. And it kind of works like this. In the spring, when it's warm, lots of sun, the phytoplankton, phyto means plant, the ones that have chlorophyll become abundant. And then as they become highly abundant, then the zooplankton will feed on the phytoplankton. They become very abundant, and when they run out of food source, they break up, disintegrate, and the bacteria are released into the water column. And then if you drink the water that has not been treated, then that's when you can come down with cholera. But it takes a lot of bacteria. It takes about cells per milliliter. And we fortunately no longer have cholera, haven't had cholera for probably since 1930, when chlorination was introduced by the team at Johns Hopkins to the country. So cholera now is for the developing countries, but it's global. You see in Africa is where cholera is now predominant. In Latin America in 1990, 1991, during the El Nino years, cholera, especially in Peru and Mexico, occurred. But as I said earlier in the introduction, cholera has been originated in Asia, but that's simply because that's where it continues to be a serious problem. We're moving now into Bangladesh, and this is the Gangetic Delta, and those are the Himalayas. The relationship between the Himalayas is very important. I've done most of my work in this area called Matlab. It's east of and most of the work is done in the villages that surround these ponds. And so the work that we have done has been over the last 40 years tracking cholera in the natural environment. And I'd like to point out the relationship to chlorophyll is important. Here is starting in 2003, 2004, 2006, 2007, you can see the cycle of plankton around the coastal areas of the major continents of the earth. And it's usually in the coastal areas that this disease becomes dominant, and it's cyclical, spring and fall. And remember the cyclical nature, because I'll have something to say about that when we get to discuss the coronavirus. Now, working in Bangladesh these years, it occurred about 20 years ago that if chlorophyll was so important, and the Landsat satellite had been launched in 1972, and by 1980, we were able to measure by satellite chlorophyll, sea surface temperature, sea surface depth, and it occurred to our team at Maryland that we could use satellites to predict cholera outbreaks. And so that's what we set ourselves up to do. We, in fact, carried out a series of experiments. Let me go back, I think. A series of experiments. Now, so you can see the problem here pretty easily, where in a country like Bangladesh, where water is fetched from a pond, no treatment, people are washing clothes, brushing teeth, collecting water for drinking. The transmission is easily done. So in the work in Bangladesh, we were able to confirm what we had discovered in the Chesapeake Bay, because we had been criticized that, what happens in the Bay doesn't really mean that's what happens around the world. Well, the work in Bangladesh that we began in 1975, we found that indeed we could pick up the bacteria in the environment. Obviously, with the sanitation problems, as I just showed you, there's person-to-person transmission, but fundamentally, cholera is from the environment. Now, why do I make this point? And that's because physicians and medical doctors were absolutely convinced until 20 years ago that cholera was transmitted only person to person, and you had to have an infected carrier to transmit it. But we were able to show, and it's now textbook, that the cholera bacteria are naturally occurring in the environment. They're part of the natural system. And when we drink water that hasn't been treated, hasn't been filtered, we can get cholera. Now, let me take you back to the Bay of Bengal and the satellite story that I would like to tell you. We learned from NASA and teamed up with NASA to get the data on chlorophyll, sea surface temperature, sea surface height. And it was really uncanny. This was very exciting. This was 20 years ago. We were able to show that with the Landsat, the blue line is the sea surface temperature, and the red line is cholera. And this is very crude. This is essentially counting patients coming into the hospital in Bangladesh and matching it up with satellite observation of the sea surface temperature. And similarly, we were able to show chlorophyll and sea surface height. All of this allowed us to build a model of prediction. And we use this now to predict cholera in Calcutta, in India, and in Matlab, in Bangladesh, near Dhaka. And the model is really very powerful. With every milligram per cubic meter increase in chlorophyll in the water, you'll get 33% increase in the number of cholera cases. With every millimeter per day increase in rainfall, we predict, and we can show 7% increase in cholera cases. That's in Calcutta. The same thing in Matlab, that is Dhaka, Bangladesh. So now we've taken what we've learned in the Chesapeake Bay, build a model, been able to show the relationship and detect and predict by satellite. Now we went back to the old data in the library in London that were collected by the British colonial soldiers and the diplomats who worked in India. We went to the records that they kept, and we were able to show a very definitive relationship with temperature for the kinds of epidemics that occurred inland, because what I've just been talking about now is mainly coastal water related. But with the data from India from 1870 to 1900, which we transferred to the computer and worked out relationships. This was teamwork done with Dr. Huck, my colleague at the University of Maryland, a colleague, a physician friend, Elizabeth Whitcomb, a physician from New Zealand who'd been working in London for many years. So we were able to improve our model to the point where we now have a very sophisticated model. We've used many, many more satellites. And we now know that if we have very, very hot weather, and then 4 weeks later, a very heavy rainfall, and if it's in an area of the world, like Bangladesh or Africa, where we don't have good sanitation, good water treatment, we'll have a very high cholera risk. Now, we've been able to carry out our studies. Initially, it was back in the 60s, 70s, and 80s with Landsat, mainly in the 70s and 80s. But now we use a whole series of very sophisticated satellites to build a very, very powerful model. And this is work being done, funded by the NSF, funded by NASA, and also in collaboration with the Departments of Environment and Health in Maryland. And we've been able to produce really, really sophisticated models. In fact, the models are so sophisticated that I'm showing you now Hurricane Matthew, which ravaged Cuba and Haiti a few years ago. And the path you can see of the hurricane. Now, we used our parameters to predict where in Haiti, with these conditions, hot weather, heavy rainfall, disturbed environment, and on the left, you see the prediction, and on the right, the dark red dots are the actual cholera cases. Now, this has improved to the point where in 2017, we started expanding to Africa. And this is a study we did in Yemen. We published it. And at the end of 2017, Scientific American made a little paragraph, wrote a paragraph about the work, having seen our publication. And a scientist in England working for the British Aid Agency called us on the phone and said, look, could we work with you? If you predict in Yemen where cholera will be in 2018, we'll send our physicians and supplies to the right places where the risk is highest. And so that's exactly what we did. We were able to show precisely from the data in 2015, 16, 17, and in 2018. And now we're working directly with the British DFID, the Department for International Aid, with UNICEF, the Children's UN agency, NASA, and the British Meteorological Agency. And we now provide them every month a 4-week ahead of time prediction of the highest risk of cholera in Yemen. The model has become so accurate that we actually picked up, surprisingly, this past year in 2019, risk very high in Sudan. Now, they didn't expect that, but sure enough, a month later, outbreak of cholera occurred in Sudan. So now we've been asked by the DFID and UNICEF and with NASA to continue our studies in Africa. So we're now expanding to Africa. Now let me take you to climate change. I'm trying to cover a lot of material, but this is a very interesting story as well. 40 years ago, 45 years ago now, the Southampton Marine Station in England decided that they ought to curate plankton samples and keep them just in case there would be climate change effects. So they have a record of 40 years of plankton samples stored, collected at each of those sites where you see the red squares on the North Atlantic, in the North Sea. And so what we did was go to those samples. This is work done in collaboration with Dr. Carla Pruzzo, a wonderful scientist in Genoa, Italy, and with a team at the Max Planck Institute in Germany, and with the Southampton Marine Lab, and with the University of Maryland, our lab at Maryland. Southampton Lab had all of the temperature and all of the pH and other data for the last 40 years, plus the plankton. So what we did was go in and extract the DNA from the plankton, and with very sophisticated probes, enumerate vibrios. Vibrios, Vibrio parahemolyticus, which causes seafood poisoning, and that's shown in green. Vibrio vulnificus, which is in yellow, and then the pink is Vibrio cholerae. And we were able to correlate the increase in the numbers year by year by year directly with outbreaks of Vibrio infections in England and Europe and in the New England northern area. So now we have a direct evidence of an infectious disease that is influenced and affected by climate change. As the waters warm, it becomes much more, let's say, conducive for the Vibrios to multiply. And people in England and in France who go wading on the beach or who eat shellfish that have these higher numbers of Vibrios then have these infections. Now, let me turn very quickly to genomics, because this has been very, very exciting. I've always been interested in the genetics of marine bacteria. And what I developed as a result of chairing, when I was director of NSF, a team working collaboratively of all the agencies. It was classified. We worked in a skiff, and I've written about it, and my book comes out in August and tells the story of anthrax. But we advised the CIA and the FBI using molecular genomics to track down the source. Now, what is that technique that I've since developed? We can take any sample, water, food, sewage, it doesn't matter, clinical samples, blood, urine, extract the DNA, sequence it on any sequencer, it's agnostic. And with the DNA reads that come out from the sequencer, we match it to our very elegant database with 170,000 highly curated genomes of microorganisms, bacteria, viruses, fungus, parasites. We can identify all of this right down to species and strain. And in addition, we can identify the genes that code for antibiotic resistance, pathogenicity properties, and metabolic properties. And that means we can identify the microorganisms, the bacteria, the viruses, the fungus, and the protists, the parasites, down to species and strain. Now, why do I keep saying species and strain? It's because, as you'll see in the pink box, that's our algorithm developed at the University of Maryland with students and a team. Now it's been spun out into a company called Cosmos ID, located in Rockville. And we're the best in class. All of the other, from Harvard, from let's say from the University of Washington, from Washington University, from Argonne, when we competed, we're the only ones that can really identify down to species and strain. Now, why is that important, going from kingdom down through the phylum, to the order, to the family, to the species and strain? Let me explain very briefly. Lactobacillus is the name of a bacterium that's used to make cheese, one strain. A different strain makes Chardonnay, and a third strain is used to ferment and make yogurt. For physicians, strain is important because all of us in our gut carry E. coli, Escherichia coli. It's a good bacterium, produces vitamin K, B12, it protects against pathogens. But if it goes rogue and we have a strain 015787, that causes hemorrhagic enteritis. If you eat a hamburger infected with the organism, it can be bad news. Now, let me give you an example. This was a young woman who built herself a zip line. She's on the zip line, it breaks, she falls in the water, she lacerates her calf, and this expands into an infection. It becomes flesh-eating. It's a cellular necrotizing cellulitis, flesh-eating bacteria. They looked for streptococcus, which is what usually causes the necrotizing cellular fasciitis, but they isolated Aeromonas. This is a bacterium from water. It's a water bacterium. They isolated 4 strains. We sequenced the strains. This was work done with Chris Grimm and a team at the FDA here in Maryland. And we're working with a group, Ashok Chopra at Texas, collaborative work. 3 of the strains were absolutely identical. They were clonal. 1 was different. We injected them in mice to see how they were operating. How do they cause this necrotizing cellular fasciitis? Strangely enough, we found that only 1 strain, when all 4 were injected in the mouse muscle, only 1 strain went to the liver and the pancreas, which in the case of the young woman, and these were the strains that infected her, had gone systemic. She ended up with both legs amputated to the knee and both arms amputated to the elbow. She's still alive, she survived, but she's a multiple amputee. Now, what we were able to show, because when we wrote this up, we figured that a reviewer would say, well, are you sure you're using new techniques? So what we did was engineer that 1 bacterium that went to the pancreas and the liver, and we made it kanamycin antibiotic resistant, and we also inserted genes to make it luminescent. And we repeated the experiment. And sure enough, the only strain that went to the liver and the pancreas was the luminescent kanamycin-resistant strain. So what we learned was the bacteria worked together. The strains that did not go to the liver produced a toxin that broke down the muscle tissue, and that allowed the other strain to go from the muscle tissue to the liver and pancreas. So strain identification is really, really important. And here I'll demonstrate again by studies we've been doing on the microbiome. That is all of the gut flora. That is, all of us have billions of bacteria in our intestine. And they're, thank heaven, good bacteria. They help us digest, and they protect against pathogens. And so we did a study that's now being written up with a team in Calcutta, India, where it's a cholera hospital. And when patients come in with severe diarrhea, they sent us samples of the DNA extracted from the stool sample, and then they did a whole series of tests themselves. And what we found out was that by looking at the bacteria, the viruses, and the parasites, which we can do with our identification system, we were able to show very clearly that when they knew they had a cholera bacterium, the known disease, we also picked it up. But they had sent us samples where they could not identify the pathogen causing the severe diarrhea. And we were able to show that it was in fact rogue E. coli. But then we also had some controls, always good to have controls. The healthy population. To our surprise, the healthy Indian population carried a certain number of pathogens. But then we downloaded from the NIH Human Microbiome Program, HMP program, the gut flora data for us Westerners. And we found out some very interesting things. First of all, the Indian population is more tolerant to pathogens. Secondly, we found that they also contain higher numbers of good bacteria associated with yogurt, buttermilk, and fermented products, which is a food staple in that country. But more to the point, that this approach is a very effective bioforensic tool. In other words, we have done studies now of a dozen countries, and we're able to show that what you carry in your gut is kind of, it's not a fingerprint because it's your gut, but it's a kind of gut print that, that identifies you as English, Canadian, Nigerian, Indian, Japanese. So it's a very powerful tool. And we can identify the bacteria. You can see that column down the middle, which does not have a ring around it. That's because there are almost no pathogens. And those are human Western gut flora. And then to the right and left are the Indian, to the left control, and the right, the Indian patient populations. Now, I've told you all this because I want you to understand how accurate this is. Now let's go back to water, because this is important, at least for our discussion. We've been working with Orange County, California. Now, as you know, all of us know from reading the papers, they've had severe droughts. And so some years ago, Orange County, California chose to reuse water. So water from the sewage plant is transported, just the water, the sludge goes to sea, to the drinking water treatment plant. And the treatment plant then goes through, does a series of processes, microfiltration, microfiltration, reverse osmosis, treatment, UV irradiation, chlorination, et cetera, et cetera. And so what we did was look at the samples from each step. Now you can see that clouded area. Those are all the bacteria we found in the input water coming from the sewage plant, loaded with microorganisms. Some pathogens, a few parasites as well, and viruses. But once it's gone through each step, you see fewer and fewer microorganisms until what we find by the time it's ready to be used as drinking water, we find that it's only good bacteria, the typical bacteria that you would find in safe water. That is, bacteria that convert iron or perhaps carry out some metabolic properties, but they are not pathogenic for humans. Tap water, for example, bottled water, is not sterile. I'll talk about that again in a moment. We showed the same thing for viruses. The viruses even had some human viruses in the sludge coming in, but by the time the treatment process had worked, we were able to show clearly that the only viruses were bacterial viruses, and these are not pathogenic or dangerous for humans. And another important finding, we were able to assure the Orange County water system that they were concerned that by using wastewater, they might be conferring antibiotic resistance to the naturally occurring bacteria in the water. But we found that's not the case. We found lots of antibiotic resistance in the Q1, very tall orange column. And by the time it got through the millipore filtration, still a lot. By the time it got through reverse osmosis, hardly any antibiotic resistance genes. And by the time it got to drinking water, there weren't any. So there you were. We were able to show that the water is safe. Now let me bring this together to COVID-19. This is a really nasty virus. This virus primarily affects the lungs. That's that quadrant in the upper left. It's a cross section, and it shows immune cells crowding an inflamed alveolus. And the virus breaks down the walls. You can see the wall to the upper right rather, fractionating. But it also affects the liver. And up to half of hospitalized patients have enzyme signals that indicate the liver is under difficult conditions. The immune system goes into overdrive, and the drugs given to fight the virus can be causing the damage in the liver, probably. And then it affects the kidneys, and kidney damage is common in the severe cases. And if you have kidney damage, death is more likely. It infects the intestines, and this is important too. It causes a lot of patients, 20% at least, will have severe diarrhea, but it infects the intestinal tract. And in some cases, it actually causes strokes, seizures, and inflammation of the brain. And then it can cause a conjunctivitis. It is kind of a nasty one because the inner eyelid is affected. And then the nose is also affected. Some patients lose the sense of smell. And eventually affect on the heart, because as the lung becomes damaged, then the oxygen doesn't get to the heart and to the blood vessels where it needs to be distributed to the brain. So this is a bad bacterium, virus rather. It's a bad virus, and it's one we need to respect. Now, we've been able to build our database to include the SARS-CoV-2 coronavirus, and we're able to determine variants if there are any. We've got the SARS original strain and the MERS strain in the database, and then the strain sequences that the Chinese made available and others have provided, and that's been loaded in our database. And we've done the studies in stool samples, and we're able to detect the presence of the virus. Now, why is this important? Let me just go back a minute if I can. Well, why is this important? Because we've done the studies similar to what we did in Calcutta for cholera. And now we're doing this because studies have shown that in at least half of stool samples of victims of either non-symptomatic and symptomatic coronavirus victims, the gut reveals the presence of the virus. So the virus is shed into the stool sample and into the environment. Now, this allows us then to take from the literature, this is from the literature, a very, very important study, because what it shows is that the stool samples remain positive after the lung sample or the sputum or the swab, respiratory swab, has gone negative. To explain, which seems to be a very complicated graph, it is simple when you understand that the open triangles are the asymptomatic, and the red line is the individuals that are throat swab positive, and the yellow line is the fecal sample is positive. And then the red squares when they've discharged, when they've been admitted to the hospital, and the open green square is when they've been released from the hospital. And unfortunately, the red X means death. So you can see that the throat swab will be positive in the early stages, but the stool sample remains positive and is discharged for a long time, for days, weeks afterwards. This is important because it means that we need to understand wastewater as a potential tool for detection, but also a potential transmission if it goes into the environmental system. And so the SARS-CoV-2 has been detected in stool samples of patients infected, as I've just pointed out, and studies done in the US, my colleague Chuck Gerba at Arizona, and the colleagues in Australia and in the Netherlands and in Sweden have shown that sewage carries a viral load and can be used as a monitoring mechanism for when it's in the community before the cases have shown up. So it can be used to measure how effective interventions are, social distancing, and so forth, because the numbers, as was shown in Tucson by Gerba, the numbers go down as the numbers of people carrying it go down in the community, and vice versa. As the numbers go up, the amount in the sewage will be detectably increased. Now, I'm closed because I'm running out of time, and I want to get questions from you. We've been looking at drinking water, tap water, and bottled water. And so what we did was, frankly, just go to various taps in Maryland and in DC, and we collected, we went out and bought bottled water, different kinds of bottled water, mineralized, sparkling water, still water, etc. And then we did analyses and found, of course, they're not sterile, but it's very interesting. The sparkling water, that's blue, carries mainly bacteria that you would find in wells and deep aquatic systems. The green dots are tap water, municipal tap water. I'm sorry, they're non-mineral bottled water, and off to the left is the tap water. So it just has these have a very characteristic microbial flora. Now we've looked at what's there, and we can actually characterize, just as we did stool samples, different kinds of bottled water. And then we're able to show exactly down to species and strain what's in the water. The bacteria, fungus, yes, there are bits of fungus in some of the bottled waters, harmless, and some protozoan, non-pathogenic, fortunately, and some ancient bacteria that we find in the mineral waters. And we now know that, and this has been published, it's just appeared a week or so ago, and this is the work of a very good graduate student, Kyle Brumfield, my wonderful colleague, Dr. Anwar Huq, and Joe Catruvo and Menu Letty team working together. The predominant bacterial species in water were members of Actinobacteria, Proteobacteria, harmless, good bacteria. characteristic species and strains in sparkling water and in still water and in tap water. We did pick up opportunistic plant and animal pathogens in a few samples, not human pathogens. No fecal indicator bacteria. We're drinking good water. That's always a good sign. We did pick up bacteriophages, just as I showed you, we found in that highly treated Orange County water, but these don't infect humans. In fact, they are a pretty good code for other bacteria that might be present, for which they're very specific. And then we detected some ancient bacteria only in the mineral water. So let me close by saying safe water is critical, and it is the challenge of the humanity. We've already seen oil barreling out, shall I say, to coin a phrase, but it is water that we really need for the future. All that I've told you is important because we found out that because the Vibrio cholerae are attached to copepods, these are pretty big, and they can be filtered out by sari cloth. And so in the remote villages of Bangladesh, we were able to train the women to filter their water, and with the filter, after filtering it, to rinse it off and sun dry it to disinfect it and reuse it. And we were able to provide clean water. To the right, you can see filtered water. To the left, you can see cloudy water with stuff that's still swimming. And we were able to reduce cholera by this simple technique by 50%. So the high technology tools that we used could be simplified and allowed to be used to provide a technique for villagers in remote villages in Bangladesh. I have to shout out for the wonderful people in Bangladesh at the International Center for Diarrheal Diseases Research Bangladesh, and colleagues at the University of Maryland, students, postdoc, visiting scientists, collaborators from other countries, from around the world, and especially Dr. Huck, Dr. Jutla, who's a genius on modeling, Sion Young, who's been fantastic building the database, and Nur Hassan, who's a genius both in informatics and molecular biology. But safe water is a global challenge, so I'll be happy to take any questions.
Well, thank you so much, Dr. Colwell. We have quite a few questions already coming in. So everyone, please feel free to go down to your chat box and send a question to me. I did send a note out to everyone, just reminding them to send your questions via chat. And we are going to start to go through those questions. I hope we can get to a lot of them, but we'll try to get to all of them. And I do want to just start out with the first question from Bill Ball. And you probably know Bill, don't you, Rita? But anyway, in the plot of the sea surface temperature and cholera plot over time, the correlation was indeed remarkable. But on the plot, I noticed a very obvious and clear opposite trend for the period of March to July in 1993, where there was a high sea surface temperature and low cholera. I have no doubt there were special conditions that caused that over this particular time frame, but I wonder if they were ever uncovered. Do you have the answer?
Very clever, very
sharp eye.
Sharp eye. You can join my lab any day. It turns out that was a year where the waters coming down from the Himalayas was lessened, and there were the water supply was much lower. And in fact, there were hydrologic factors that created that anomaly. And the other thing I should tell you is that is a corrected curve in the sense that the correlation with chlorophyll is not direct. It's about a 4 to 6 week lag. But what we did was to correct it, and that provided that good correlation. It's a legitimate correction. It's just allowing for that time lag from phytoplankton to the zooplankton bloom, to the release of the vibrios, to the drinking of the water, and the outbreak of cholera. And then if you corrected that, you get that direct correlation.
Okay, well, thank you. And we have also from Ben Grumbles a question And Secretary Grumbles indicated that Maryland Department of the Environment and the Department of Natural Resources are representing Maryland in a bipartisan multi-state commitment to combat ocean acidification, which is a very real and disturbing impact on the excessive levels of carbon dioxide. What are the human health implications of an acidified ocean? Thank you, Rita.
Yes, wonderful, wonderful question. I think what that does is change the aquatic flora significantly. It's a selective factor. pH will select out different kinds of microorganisms, and we may see some surprises. We may see a bacterial equivalent or maybe another viral equivalent, because there are more viruses in seawater and in Chesapeake Bay water than there are bacteria. And that was a surprise that one of my students, who is now a professor at the University of Delaware, discovered during his PhD work. So with the change in pH, we do need to know what the microbial metagenomic composition really is, so that we can determine what selection takes place and what surprises there may be there for us, to which we want to be prepared.
Okay, and we have another question here from Teresa McKenna. Is Dr. Colwell suggesting that COVID-19 can be transmitted via untreated water, or only that it can be detected in the fecal matter?
Excellent question. There's a study that's been published, or at least I have a preprint. It should be published by now, from a team at the University of California, I think University of Southern California, where they did studies of sea spray, ocean spray, when the waves were very active, in a beach area where wastewater was flowing in, and they detected the coronavirus in the sea spray. And the beaches were closed a month ago in California. Of course, the surfers didn't like that, but I think it was a good idea for them not to be surfing.
That's going to be a bit disconcerting to all of our sailors here, if that's the case.
Well, I think we do a pretty good job in treatment, but this is something we need to look at.
Most certainly. We have a question also here from Susan Johnson. Wants to know, when I go to a restaurant, when they finally open back up, should I ask for tap, bottled, or sparkling water?
Well, when I travel in a developing country, I always ask for carbonated water, and I always ask for it to be opened at the table, because the carbonation creates that low pH that Ben just asked about. And it is actually inhibitory to E. coli and fecal bacteria. But I don't think you need to worry about water in restaurants in the US, at least in the restaurants that are obviously maintaining good sanitation and are being inspected. They're doing a good job. And tap water, frankly, I think, just a personal choice, I prefer good old Maryland tap water to still bottled water, but I will take carbonated or sparkling bottled water. That's an indirect way of answering the question.
We all have our preferences, but good suggestion there. We have also a question here from Ann Powers, and I'm sure you know Ann, but are there sewage treatment plants that are testing for the coronavirus?
There are. There's a study done in Tucson, Arizona, and the study in Netherlands. In fact, in Germany, they're really adopting this as a mechanism to determine which communities are being subjected to a potential coronavirus infection. That is, if you're starting to pick up coronavirus in the water system, at least a week from now, you're probably going to have cases. So you might as well be prepared. And that's what they're doing in Germany. And I believe getting started in Netherlands as well.
Okay. Also, there's a question from Carol McGillen, is any of this testing or modeling being used that's being used to study COVID-19 and will help us with accelerating our testing models? In other words, will the testing that's modeling that's being used to study help accelerate the overall testing models?
Yes, I think it can help, and as follows. As we all know, we cannot get tested just because we want to. Their tests aren't available. But they're doing, the governors are doing their best. And I have to give a shout out to our governor. He's fighting hard for us to have access to tests. But if we don't have all the tests, this is one way of measuring a community carriage of the coronavirus. And the numbers, and we can quantitate this, can give us an indication either of an increasing trend or a decreasing trend, or even the presence thereof. Let's say a rural community that has no cases, they want to know, should they be worried? Well, let's look at the sewage treatment plant where their sewage goes into, the plant that their sewage goes into, and determine if we can detect large numbers. If we can, then we want to watch out. So I think we can use it intelligently, not as a magic marker, but intelligently to work together with the policymakers, the managers, the governors, the staff, the wonderful staff in our Maryland administrative teams, and give them that information that they can incorporate, and it gives them greater confidence in the decisions they make.
Well, speaking of our administrative staff here in this great state of Maryland and Governor Hogan, Secretary Grumpels has an additional question for you. How can Maryland take the lead in the race to test for pathogens in sewage and water to prevent future epidemics?
Just team up with us, then we'll do it.
You'll work as a team player there.
You bet.
But I will, I know, while we have Ben on the line as well, I do want to just say that we do have a great governor, as you said, Dr. Colwell. I think he is truly listening to the scientific community, and that is very commendable. And I want to throw in a question from me, given that comment. Is it in your report that you just published in 2019, Science During Crises? I read that report. It was compelling to me that we have a strong need for action at all governmental levels to ensure that science is folded into the decision process, and that we're not opening up too soon or closing too soon, various facilities. What do you have, or could you perhaps summarize very briefly sort of some key takeaways from that major study that was done by the Academy and under your leadership?
The major message is that every governor should have a chief science advisor, just as the president has an Office of Science and Technology Policy. And this can be extremely helpful because when there is a major catastrophe in a given state, then the governors and their science advisors can work together as a team collectively and assist the given state that is having, whether it's a hurricane that's going through, or tornadoes, or the pandemic, or whatever. It can be a variety of catastrophes that occur. But having the best science available and at the ready to be able to come in and call as your scientific troops, so to speak, it empowers, I think, the governors and the country. And so that was the major recommendation. We made a series of other recommendations. For example, right now, the various agencies cannot share data. That doesn't make any sense whatsoever. And that requires a simple change by Congress that allows data exchange and sharing so that this massive data amongst all the agencies could be pooled in this time of of machine learning and artificial intelligence, where you sift through the data to find out things that can help you manage much better. So that was another major finding.
Very important. We have a question here from Mark Trice with Department of Natural Resources. Are there models estimating the possible increase in Vibrio and related infections in the Chesapeake Bay due to climate change?
Now, the answer is very resoundingly yes. In fact, Dr. Huck and the team, which I participate with, are funded by the National Science Foundation to develop specifically a Vibrio prediction model, sort of like a pollen count, so that we can know where and when in the Chesapeake Bay it would be not a good idea to go swimming, or if you do, to be very well aware if you get any kind of abrasion to be treated promptly and avoid the risk of Vibrio vulnificus, which can be a real baddie.
Yeah. Another question here from Bill Ball. I would hope that chlorination of wastewater would kill coronavirus. In this regard, I note that when this technique is used in wastewater, it is usually, and always in this region, followed by dechlorination. This is done, for example, at the Baltimore Back River water treatment plant. It's much yet known about the effect of chlorine on the coronavirus in water and the times and doses required.
That's an extraordinarily good question, because of the suggestion of chloroquine as a potential pharmaceutical treatment, which has turned out not to be either really effective and in fact dangerous. It causes heart failure in unfortunately too many of those who have been treated with it. The question of chlorination per se is a good one. The interesting thing is the coronavirus is bounded. It has a coating that's a lipid coat. So I think more likely, if we could figure out a way to introduce some sort of lipid digester that would be safe, it might be something like Metamucil, where you dissolve the lipid, and that would be a better and more effective agent at the end of the chlorination steps, rather than depending on the chlorine itself. But we're flying this airplane, this coronavirus airplane, while we're building it. We're just learning as we go along. So I wouldn't take what I've just said as the definitive statement about chlorination, but my hunch is that we would want to use some other kind of treatment that would directly affect the virus.
Dr. Ball also had an additional question, follow-up question with regard to this. He asked if you were familiar with NOAA's Vibrio vulnificus prediction tool that is online.
I am, and I am delighted that the work they're doing is excellent. And I've done collaboration. In fact, I chaired a committee several years ago for NOAA to establish a microbial component, which I think they've now got going. So yes, and we really need to team up even more because the folks there are terrific.
Okay, and I have also a comment, not a question, but from Elvia Thompson saying this is of Annapolis Green, commenting that your remarks are very, very interesting and very much appreciated your remarks. And wanted to just also query, perhaps given we're all here in the Chesapeake Bay area, could you perhaps elaborate a little bit further on the relationship of climate change to the health of our Chesapeake Bay region? I know you've pointed out, and we've been discussing sewage treatment plants and some of the issues regarding to COVID, but in terms of the the general, what you're seeing right now in the Bay as a result of climate change and human health.
I think one of the sad effects will be the disappearance of Tillman Island and our wonderful um, natural resources that the sea level rise will affect. That's one. The other is the potential for severe storms. I sure hope we don't become a tornado alley like Tennessee and Alabama. Um, but the prediction for more and more severe storms, heavier hurricanes, that's going to have a huge impact on human health. And then I think the effect on our wonderful Chesapeake Bay, the question that Ben Grumble mentioned on acidification could really affect our fisheries significantly. And we have the best fish resources in the world, really. I'm biased, but that's okay. And so I think we'll see shifts in populations, and perhaps some of our favorite fisheries will decline even further. So there will be changes, and on this Earth Day, this 50th anniversary, we really should turn our attention to how to reverse the changes that are being made, and at least, if we have to, consider how we can attenuate or adapt to the changes. I hope it's not adaptation, because those may very well be very severe. But let's be hopeful. Let's be hopeful that we can, in fact, understand better what we can do and get it started and get it done.
Good point. I think all of us, at least in this audience, clearly support efforts to adapt and to try to mitigate the impact of climate change on our, so not only on our own backyard here in the Chesapeake Bay, but around the world. I have an additional question here from Bill, also as well, from Bill Ball. He wanted to say a huge thanks to everyone involved, and especially to you, Rita, for taking the time to give us an update and engage us in a discussion. And we also want to just say, give you a thanks of appreciation from the Chesapeake Bay for everything you've done in the Chesapeake Bay region. I'm sure a lot of folks aren't aware of the fact that you and I met way back in the 80s.
That's right.
When I was the director of the EPA Chesapeake Bay program, and you were the Sea Grant director for Maryland. And my gosh, Maryland did some phenomenal work in the Chesapeake Bay and continues to do so.
Exactly.
And, so I know that certainly Bill feels that way as well. We really appreciate everything you have done for the Bay and for the world. And we also have some more questions still coming in, but I have to just tell you, there are just a series of thank yous and from a range of different folks coming into me right now saying thank you so very much for an incredibly interesting and very informative talk. We so appreciate everything you're doing, and a huge thanks to you and for everything you've done. I can't begin to tell you how many of these I'm getting in right now. But seriously, we really appreciate your time tonight, Rita, and I know all of us care very passionately about the Bay and about our world. And I know that also one of the things that I just want to comment what our Commodore said today on a post was that every day is Earth Day at Eastport Yacht Club. We all care very much about this special place we call Earth, our home. And one of the things that I also know that, in fact, I'll pick on Ben Grumbles. Ben once commented at one of the presentations he made at the Eastport Yacht Club that Eastport Yacht Club, the EYC stands for Environmental Yacht Club. And, and we truly do feel that the club is very committed to trying to do the right thing. And I think you have in this audience and also in our community partners from the Bay Area that are with us tonight, you have a force of, we have a force of nature here that can really make a difference. All of us can, I think, by working together. And we do need to have that science, that accurate science to support our efforts and to help us advocate our causes. And thank you again so much for all of your time and effort. And we so very much appreciate your spending the evening with us on this very special 50th anniversary of Earth Day. My gosh, it really is amazing what has happened in the last 50 years and what's lays ahead. We really need to take action now to ensure that our future continues in a positive manner. But thank you again, Dr. Colwell.
Thank you. Thank you. It's been terrific. It's been great fun. And I'll expect to meet you at the bar at the Yacht Club in the future when we get back to normal.
I owe you a drink. Thanks again.
Good. Thank you. Cheerio.
We're going to close off
Very good.
the meeting. Thanks again. Bye, all.